Welcome everyone. I'm excited to kick off our presentation on the respiratory system. Today, we'll explore how this vital system keeps us alive and breathing. We'll cover its main components, how it functions, and why it's so important for our health. So, let's get started and discover the incredible world of our respiratory system. Now, let's take a closer look at the disorders of the respiratory system, which can affect our ability to breathe effectively. Moving on, we'll explore how gases are transported within the body, a vital process that ensures oxygen reaches our tissues and carbon dioxide is expelled. Then, we'll discuss respiratory volumes and capacities, which are essential measurements that help us understand lung function and health. Next, we'll examine the human respiratory system as a whole, including its structure and how it supports respiration. Following that, we'll look into the regulation of respiration, which involves the nervous system controlling our breathing rate and depth to meet the body's needs. Then, we'll cover the exchange of gases, the critical process where oxygen moves into the blood and carbon dioxide is removed. After that, we'll discuss the mechanism of breathing, how our respiratory muscles work together to facilitate inhalation and exhalation. Finally, we'll identify the key respiratory organs that make all of these processes possible. Throughout this presentation, our goal is to understand how these components connect to keep our respiratory system functioning smoothly and to recognize potential issues that can arise. Let's take a closer look at the respiratory organs used by different organisms. In terrestrial animals, we see vascularized bags called lungs, which are responsible for pulmonary respiration. These lungs are specialized structures that facilitate efficient gas exchange between the air and the blood. Moving to aquatic animals, most of them, like many crustaceans and mollusks, use specialized vascularized structures known as gills or branchial respiration to extract oxygen from water. Insects, which are also terrestrial but have a different approach, rely on a network of tubes called tracheal tubes. These tubes directly transport atmospheric air within their bodies, bypassing the need for blood to carry oxygen. Then, if we look at earthworms and similar invertebrates, they use their moist cuticle to absorb oxygen directly from their environment. Simple, but effective, because their body surface remains moist to facilitate diffusion. Finally, for the most primitive invertebrates like sponges, coelenterates, and flatworms, gas exchange occurs through simple diffusion across their entire body surface. Because their bodies are small and thin, oxygen and carbon dioxide can diffuse easily without specialized organs. All of these different structures highlight how diverse life has evolved various adaptations for respiration, depending on their environment and body design, which ties back to our overall theme of understanding how organisms meet their oxygen needs. Now, let's explore how gases are transported by the blood. First, oxygen is utilized by our cells to carry out vital catabolic reactions, which produce carbon dioxide as a waste product. This CO2 then needs to be transported back to the lungs for removal. Moving on, the larynx plays a crucial role in voice production and airway protection. Gases like oxygen and carbon dioxide diffuse across the alveolar membrane, which is essential for gas exchange in the lungs. This diffusion also occurs between the blood and tissues, ensuring that oxygen reaches our cells and CO2 is carried away. Breathing, or pulmonary ventilation, is the process where atmospheric air is drawn into the lungs and the CO2-rich alveolar air is expelled out. The bronchi serve as major air passages, guiding air to and from the lungs. Now, respiration involves several steps. The thoracic chamber forms the protective enclosure around the lungs, which are situated within this airtight space. The site of actual gas exchange is in the alveoli, tiny sac-like structures, where oxygen and carbon dioxide diffuse between blood and air. The conducting part of the respiratory system transports air from outside to the alveoli, passing through the trachea, bronchi, and bronchioles. The alveoli and their ducts make up the respiratory or exchange part. The pharynx starts at the external nostrils and continues down to the terminal bronchioles, serving as the conducting pathway. The lungs are wrapped in a double-layered pleura with pleural fluid in between, which reduces friction during breathing. The branching network of bronchi, bronchioles, and alveoli forms the structure of the lungs. Each terminal bronchiole leads to multiple alveoli, which are small, thin-walled, and richly vascularized, perfect for gas exchange. The trachea and primary, secondary, tertiary bronchi, along with initial bronchioles, are supported by incomplete cartilaginous rings that keep the airways open. As the bronchi subdivide, they form secondary and tertiary bronchi, ending in the very fine terminal bronchioles. During swallowing, the epiglottis acts like a lid, covering the glottis with a thin elastic cartilage flap to prevent food from entering the larynx. All these structures work together seamlessly to make sure air reaches the alveoli efficiently, enabling effective gas exchange, an essential part of respiration that keeps us alive and functioning. Now let's talk about the mechanism of breathing, focusing on how our lungs actually expand and contract. When we breathe out or expire, the intrapulmonary pressure rises slightly above the atmospheric pressure. This pressure difference causes air to be expelled from the lungs. It's like when you squeeze a balloon, forcing the air out. Then, when the diaphragm and the intercostal muscles relax, they return to their normal positions. This relaxation decreases the thoracic volume and, consequently, the pulmonary volume. As the volume decreases, the intrapulmonary pressure rises above the atmospheric pressure again, pushing the air out. On the flip side, during inspiration or inhaling, an increase in pulmonary volume occurs. This expansion lowers the intrapulmonary pressure to below the atmospheric pressure, creating a negative pressure inside the lungs. This pressure difference causes outside air to rush in, filling the lungs. A key player here is the intercostal muscles. When they contract, they lift the ribs and sternum, expanding the thoracic cavity further. This increases the volume, facilitating inspiration. And of course, the initial step in inspiration is the contraction of the diaphragm itself, which pushes downward and enlarges the thoracic chamber even more. So, in essence, inspiration kicks off when the intrapulmonary pressure falls below atmospheric pressure, allowing air to flow in. Conversely, expiration happens when the pressure inside the lungs exceeds the outside air pressure, forcing air out. Remember, breathing involves these two stages, drawing air in during inspiration and releasing it during expiration. Both rely on subtle pressure changes within the lungs, driven by muscle movements and volume adjustments, which are crucial for efficient gas exchange and overall respiration. Let's take a closer look at the total volume of air the lungs can hold at the end of a forced inspiration. This includes several components: the residual volume, expiratory reserve volume, tidal volume, and inspiratory reserve volume. When you add all these together, you get what's called the total lung capacity, or TLC. Essentially, TLC represents the maximum amount of air a person can hold in their lungs after a full, deep breath. Moving on, the total lung capacity also includes tidal volume and expiratory reserve volume. This gives us an idea of the maximum volume of air a person can expire after a normal, relaxed inspiration. It's a key measure in understanding lung function. Next, we have the expiratory capacity, which is the additional air you can forcibly expire beyond a normal exhalation. On average, this is around 1,000 to 1,100 milliliters. It's useful for assessing how much extra air your lungs can expel during forced breathing. Then there's the expiratory reserve volume, the maximum amount of air you can breathe out after a full, forceful expiration. This includes the expiratory reserve volume itself, plus tidal volume and inspiratory reserve volume, representing the total air you can forcibly exhale. Vital capacity is another important concept. It's the total amount of air you can inspire after a normal expiration. Think of it as the maximum amount of air you can move in and out with deep breathing, combining tidal volume and inspiratory reserve volume. In addition, we have the inspiratory capacity, which is the volume of air you can inspire after a normal exhalation. It includes the expiratory reserve volume and the residual volume remaining in the lungs after exhaling normally. The inspiratory reserve volume is the extra air you can breathe in with a forceful inspiration, averaging around 2,500 to 3,000 milliliters. This capacity allows us to take deeper breaths when needed. Moving to the functional residual capacity, this is the volume of air left in the lungs after a normal exhalation. It includes the residual volume plus the expiratory reserve volume, typically around 1,100 to 1,200 milliliters. The residual volume itself is the air that remains in the lungs even after maximum exhalation. It's about 500 milliliters and ensures our lungs stay partially inflated, preventing collapse. Finally, tidal volume is the amount of air inspired or expired during normal, relaxed breathing, usually around 500 milliliters. It's the breath we take effortlessly, forming the basis of our respiratory activity. Understanding these volumes and capacities helps us evaluate lung health and function comprehensively, connecting the mechanics of breathing to overall respiratory efficiency. Let's talk about the pressure contributed by individual gases in a mixture. This is called partial pressure, and it's a way to measure how much of each gas is exerting pressure within the mixture. For example, we use PO2 to represent the partial pressure of oxygen and PCO2 for carbon dioxide. Now, these gases don't stay put. They're exchanged mainly through simple diffusion. This process happens because of pressure or concentration gradients, meaning gases move from areas of higher partial pressure to areas of lower partial pressure. This gas exchange isn't limited to the lungs alone. It also occurs between the blood and tissues throughout the body. The primary sites where this exchange takes place are the alveoli, tiny air sacs in the lungs. These alveoli are incredibly efficient at facilitating the transfer of oxygen into the blood and removing carbon dioxide from it. Understanding this exchange of gases is crucial because it underpins how our respiratory system supports cellular function and overall homeostasis. So, when it comes to carbon dioxide transport in the blood, about 7% is carried dissolved in the plasma. That may seem like a small amount, but it's still important to recognize. A larger portion, nearly 20 to 25%, is transported by red blood cells, or RBCs. The majority, around 70%, actually travels as bicarbonate ions, which forms when CO2 reacts with water inside the blood. This process is crucial because it helps regulate blood pH and efficiently remove CO2 from the body. Now, shifting to oxygen transport, only about 3% of oxygen is dissolved directly in the plasma again, a small fraction. The vast majority, approximately 97%, is carried by RBCs. This highlights how vital red blood cells are for oxygen delivery. They contain hemoglobin, which binds oxygen tightly and allows efficient transport throughout the body. Understanding these proportions really emphasizes the sophisticated way our blood manages gas transport, balancing between dissolved states, cellular carriers, and chemical forms to keep our tissues supplied and our blood balanced. Let's talk about oxygen transport and how hemoglobin interacts with oxygen in different conditions. Every 100 milliliters of oxygenated blood can deliver around 5 milliliters of O2 to the tissues under normal physiological conditions. Now, the binding of oxygen to hemoglobin is influenced by several factors. First, consider the oxygen dissociation curve, which is a sigmoid curve that shows the percentage saturation of hemoglobin with oxygen plotted against the partial pressure of oxygen, or PO2. This curve is incredibly useful because it helps us understand how various factors like CO2 levels and pH affect hemoglobin's ability to carry oxygen. In the lungs, or alveoli, where oxygen is abundant, the conditions favor oxygen binding. Here, the PO2 is high, and hemoglobin tends to be saturated with oxygen. Conversely, in the tissues, where oxygen is being used, the conditions favor oxygen release. There, the PCO2 is higher, pH is lower, temperature is increased, and PO2 is lower, all of which promote oxygen dissociation from hemoglobin. Specifically, higher temperature and high PCO2, along with increased H+ concentration, make it easier for hemoglobin to release oxygen. On the other hand, in the tissues, lower PCO2, lesser H+ concentration, high PO2, and lower temperature favor the formation of oxyhemoglobin, helping hemoglobin pick up oxygen when needed. It's important to remember that each hemoglobin molecule can carry up to 4 oxygen molecules, making it a very efficient carrier. Hemoglobin itself is a red pigment containing iron, and it's present in red blood cells, which are crucial for oxygen transport. In summary, the binding and release of oxygen depend heavily on these factors, and the sigmoid shape of the curve visually represents this dynamic process. Understanding this helps us see how the body efficiently loads oxygen in the lungs and unloads it in the tissues, adapting to varying physiological conditions. Let's talk about how carbon dioxide is transported in the blood. For every 100 milliliters of deoxygenated blood, about 4 milliliters of CO2 are delivered to the alveoli. This CO2 is mainly trapped as bicarbonate at the tissue level and then transported to the alveoli, where it gets released as CO2. At the alveolar site, the process reverses. CO2 and water are formed again, allowing CO2 to be exhaled. Meanwhile, at the tissue level, CO2 diffuses into the blood and reacts to form bicarbonate and hydrogen ions. This reaction is facilitated by an enzyme called carbonic anhydrase, which is found in high concentrations in red blood cells and in smaller amounts in plasma. When the partial pressure of CO2 is low and oxygen is high, such as in the alveoli, CO2 dissociates from hemoglobin. Conversely, in the tissues, where CO2 levels are high and oxygen is low, more CO2 binds to hemoglobin to be transported back to the lungs. In fact, about 20 to 25% of CO2 is carried by hemoglobin as carbaminohemoglobin. So, this system allows efficient CO2 transport, balancing the needs of tissues and the lungs, and maintaining overall respiratory function. Now, let's explore how our body monitors and regulates respiration to keep everything in balance. Receptors located in the aortic arch and carotid artery play a crucial role here. They can detect changes in carbon dioxide levels and hydrogen ion concentration in the blood. When they sense these changes, they send signals to the respiratory rhythm center, prompting the body to make the necessary adjustments to maintain proper gas levels. Additionally, there's a part of the brain called the pneumataxic center, which is located in the pons region. Its job is to moderate or fine-tune the activity of the main respiratory rhythm center. Think of it as a regulator that helps smooth out our breathing rhythm, ensuring it remains steady and appropriate for our body's needs. At the core of this system is the respiratory rhythm center itself, situated in the medulla region of the brain. This center is primarily responsible for controlling the basic rhythm of breathing, setting the pace, and ensuring our respiratory patterns are consistent and responsive to our body's requirements. Together, these components form a sophisticated feedback system that constantly monitors and adjusts our breathing, highlighting how finely tuned and responsive our respiratory regulation truly is. Now, let's talk about long exposure to harmful substances in the workplace. When workers are exposed to dust or fumes over a long period, it can cause inflammation in the lungs. This inflammation can lead to fibrosis, which is the proliferation of fibrous tissues in the lungs. Essentially, this means the lung tissue becomes thickened and scarred, leading to serious lung damage. To prevent this, it's crucial that workers in such industries wear protective masks to reduce their inhalation of hazardous particles. Moving on, in certain industries like grinding or stone breaking, there's a significant production of dust. This dust is a major concern because it can be inhaled and cause various respiratory problems over time. One such problem is emphysema, which is a chronic disorder. In emphysema, the walls of the alveoli, the tiny air sacs in our lungs, get damaged. This damage reduces the surface area available for gas exchange, making it harder to breathe. A key cause of emphysema is cigarette smoking, which dramatically increases the risk. Lastly, let's discuss asthma. This condition makes breathing difficult and often causes wheezing. The main issue in asthma is inflammation of the bronchi and bronchioles, which are the air passages in our lungs. This inflammation narrows the airways, making it hard to get air in and out, especially during an attack. All these disorders highlight how important it is to protect our respiratory system, whether through workplace safety measures or lifestyle choices, to maintain healthy lungs and overall well-being.